IMOP in Poland. A few of us met Gregory, the president of the French Society of Parassitology, and we decided to be joined in several initiatives as this webinar. So this round table, this joint meeting between the French and Italian Societies of Parassitology as a theme, transboundary parasites, and we think that it is very timely. And I hope that this will be only the first of a series of webinars we can organize in the near future. Okay, so after a brief presentation of the Italian Society of Parassitology by myself and the same for the French Society of Parassitology by Gregory, we are going to present some themes on transboundary parasites by Adriano Casulli, by Alessandro Massolo and Gerard Humag, and this first section will be based on echinococcosis. And then Simonetta and Melanie prepared a presentation on fish parasites of zoonotic importance. And then I hope we will have a discussion about these two topics. So SoIPa is the Italian Society of Parassitology. It was founded in 1959 in continuity with the Society for the Study of Malaria, already established in 1898. We have several missions, so to strengthen collaboration among researchers and practitioners of parasitology and parasitic diseases, to promote parasitology research, to promote also dissemination and correct information on different parasitological issues, but also to support the teaching of parasitology in across Italy at both academic and non-academic level. The executive board is pretty new. We were elected last year in Padova. Really, I think it's a dream team because we have a very good relationship. So I thank really the Vice President Adriano Casulli, the Treasurer Emanuele Brianti, but also all the board members, David Di Cave, Sara Epis, Valentina Mangano, Antonio Varcasia, the past president Fabrizio Bruschi, and the secretary Maria Paola Maurelli. As I said, research is important, dissemination is important for our society, but also teaching, promoting teaching of parasitology in the vet school, in the medical school, in the biology school, in a multidisciplinary way, it's important for us. And this is why we also have a teaching committee with more than 80 professors of parasitology and parasitic diseases of animals and humans. And this team is coordinated by Simonetta Mattiucci. And so our aim is to promote the teaching of parasitology across Italy at a different level. SoIPa has 276 members belonging to different fields, so veterinary fields, medical fields, biology and ecology fields, but also other fields because, again, multidisciplinarity is very important to us, and now we have new members, even environmental engineers as new members of our society. The topics are different. I think this is the same also for the French Society. So we deal with parasites of farm animals, companion animals, exotic animals, wildlife, fish and aquatic animals, vectors, but also mycotic diseases, diagnosis, drug resistance, and also alternative methods for parasite control. We are in partnership not only with the French Society of Parassitology, but we are federated with the World Federation of Parasitologists. We have an important role in the World Association for the Advancement of Veterinary Parasitology, WAVP. We are part of the European Federation of Parasitology. And at national level, we are in partnership with the Italian network of for NTD, with the Federation of Veterinary Sciences, and with the different associations dealing with microbiology and also tropical medicine. Our Congress is biannual since 1959, and our last Congress was organized by the colleagues Rudy and Antonio in Padova, and was really a great success. And Radu, this is a video. Okay, so the next Congress will be organized by the colleagues in Bologna. The theme is cooperation in parasitology, and we have the dates from 23 to 26 June 2023. So, as I said, the theme of our next Congress organized by the colleagues from Bologna is cooperation in parasitology, and it will be from 23rd to 26th of June next year. So I hope we can really organize something together. Dissemination activities, press release, videos. We have also people will, and board members on TV promoting parasitology, news, newsletter, webinars, and training videos. As I said, we promote parasitology, especially for young researchers, and we have several initiatives, for example, the SoIPa Awards, Spring Parasitology, which is a webinar series dedicated to the presentation of PhD and postdoc projects by young researchers, and also the Youth Initiative for NTDs in Italy. We also promote different on-site courses as the PARSCO. I think that you all are aware about this. Continuing education courses, for example, the latest one was organized in Toscana and was dedicated to veterinary and human leishmaniasis. And we are in partnership with different associations in order to co-organize congresses beyond SoIPa. And as was announced in Poland last year, the colleagues from Rome, from the University of Rome Sapienza, Istituto Superiore di Sanità, and Tor Vergata, will organize the next IMOP 2028 in Rome. So I'm sure that we can co-organize some session together within this Congress. We are also on the social, so we have a website on Facebook, on YouTube, and on Instagram, and that's it. So I think we have a long tradition of collaboration with our French friends, and as I said, I hope that this will be only the first of a long series of webinars, but also meeting in presence. Ok. Ciao Simonetta.
Hello.
So maybe
Ciao.
Gregory, ciao. You can present
Thank you, Laura.
French, yeah.
So yes, as Laura said, we met during the last EMOP in Poland, and we discussed about the collaboration we could have between our two societies. So I will share my screen to present, to introduce our French Society for Parasitology, but I think most of you already know Okay. Can you see my screen?
Yeah.
Okay,
Yes.
perfect.
Yeah.
So, the French Society for Parasitology was created in 1962, so 3 years after the Italian Society. And it was at the National History Museum of Paris, and it was official in June. 4 people decided to found this society, Jacques Callot, Jean Biguet, and one of my mentor, Alain Chabot. I had just the chance to start my parasitology with Alain Chabot, Professor Alain Chabot, you see on the right, and Claude Dupuis. At the very beginning, there were 25 members. And the idea to create this society was because, of course, the parasitology in these 16 was in booming and in France and all over the world. And there was the first announcement of ICOPA meeting. It was the first ICOPA meeting taking place in Roma. So already the Italian people organizing the parasitology in September 1964. At the first executive board, the president was Professor Robert Dolphus, and the general secretary was Alain Chabot. And now, in 2025, the executive board is about 20 members. So I'm the president. Frédéric Grenouillet, which is connected to, is the general secretary. The treasurer is Émilie Boussira. I think most of people from the Italian Society know Émilie because she's a veterinarian. And the vice presidents are Christelle Pomares and Michael Rio. Our past president is Isabelle Villena. You may know her also. And there are 15 other executive committee members that you can see here on the screen. For example, Mélanie and Gérald will present today in the joint meeting. Still, the French Society now, we are 219 members. Almost half are involved in human medicine, and the other half is involved in other fields. And contrary to the Italian Society for Parasitology, there is only 8% of veterinary people, but this number will increase, I hope, in the near future. Our main activities, so of course we have a journal that you may know, Parasit. I will speak a bit more on this journal later. We organize webinar, bimensual, and we will share with your society also the dates, so you can participate to this webinar. You are really more than welcome. We organize each year a call for financial support for Master 2, 1 per year. And we have 2 prizes per year, one for PhD thesis and one for veterinarian, pharmacian, or physician thesis. We have an annual meeting. I will come back a bit later on this for this year. And we have active participation of our members to the European Federation of Parasitology and the World Federation of Parasitology, including in EMOP and ICOPA. We also are present in different social media, such as X or LinkedIn or etc. And I just gave some ideas of future developments of the society to make some link with other parasitology societies, such as SoIPa, but also the African Society of Parasitology, the BSP, and others. We have already strong connections with French Society of Medical Mycology, and we also have more and more connection with the French Society of Microbiology. We want to increase also the sponsoring because for now we don't have so much sponsors to help for the prices, but we need many more sponsors to organize more meetings. And we want to increase also the visibility on social media. Recently, we started an account on LinkedIn, thanks to Frédéric Grenouillet. And it seems it's working very well. We have a lot of new connections now. So I think it's a good way. So I spoke about earlier our journal, Parasite. So it covers all aspects of parasitology, human, veterinary medicine, entomology, integrative taxonomy, etc., etc. It's published by EDP Science. The editor-in-chief is Jean-Loup Justin, and the associate editors are Frédéric Grenouillet, Emmanuel Liénard, and Stéphane Picot. This journal is also present on X, Blue Sky, Mastodon, LinkedIn, and on Science Open. And a lot of researchers follow the journal all over the world, mainly American people, which is very interesting because our journal is really visible now all over the world. The impact factor is stable. You can see on the left that it was around 2 in 19, and now it's still around 2 at 2.4. So it's very, very, very stable. The total citations is also stable, and the impact factor in 5 years. So we are very happy of the job done by all the editors of Parasite. If we want to just keep the key numbers, you can see that we have more and more articles published, a stable impact factor, more and more downloads of our articles, and the site score stable too. So our annual meeting last year, it was in Angers. This year it was in Amiens. It will be in Amiens in June. And of course, you are all cordially invited to participate. Slots are still available. You can look through the online pre-program and find the different sessions in which you can apply some salary. As you can see, we are very strongly joined with the French Society of Mycology, and our meeting is all years joined with this society. So the first day will be dedicated to the mycology, and the 2 last days will be dedicated to parasitology. And on the morning of the first day, so the 19th of June, it's a common session about tools in mycology and parasitology. And you can see the link just down to the slide. And as you said, I was pretty sure that Laura will introduce the EMOP because it's in Roma in 2028. And I chose to introduce the ICOPA, which will be next year in Montreal. So the dates are in August 2026. And we'll cover, of course, all aspects of parasitology. There will be experts from all over the world. And you can organize easily numerous symposia. So you can propose them and organize them. And I'm pretty sure the local organizer will be very happy to help organizing this symposia. If you want, I can make the link. I am also on the board of the WFP, and I will be involved in a scientific committee. So if you need help, just contact me, and I can help you too. Now it's time for our first SoIPa SFP joint meeting. I just remind the program, but you saw it at the very beginning, and I think let's go for a keynote. Stop the sharing. Okay, so I think if we have questions, discussions around the societies or how to work together, we can keep it for the end of this meeting. And Adriano, let's start.
Can you hear me?
Yes.
Okay, good afternoon from Rome. I will start sharing my screen. And can you see?
Yes.
Yeah.
Okay,
Yes.
good. So today my talk will focus on human alveolar echinococcosis and just briefly to remind you what alveolar echinococcosis is a neglected progressive infiltrative proliferation of parasitic small vesicle, few millimeters in length, with usually a central necrosis mimicking metastasizing tumor. And this will be a short presentation on a long study that has been conducted by the contribution of many colleagues. And the study aims to unveil the epidemiological impact, providing a quantitative measure on numbers, incidence, and trends of human alveolar echinococcosis during the last 27 years in Europe. And methodology was the same as we published a couple of years ago on cystic echinococcosis in Europe. So this is the first slide, the first picture on the number of cases during the considered period from 97 to 2023. And these are the number of cases, increasing number of cases each year that we counted in Europe. It is a steady increase of numbers, with the exception of probably this 2 years period that is probably due to the impact of COVID pandemic on surveillance in Europe on alveolar echinococcosis. And here you can see also the index cases. So the first year when country detected a case, autochthonous or not autochthonous case in Europe. So it seems to me a clear message, at least on the documented number of cases. The same pictures accounting for the period on number of human cases in Europe. Dark red means at least 800 human cases documented in Europe. During the period, we identified more than 4,000 human cases, and with a fatality rate during the period of 10%, but please keep in mind that we know that only 3% are related, the fatality is related to alveolar, and it is basically unknown in 4.6% of this cohort. You should also keep in mind that only 7 countries documented 90% of human cases. Austria, France, Germany, Switzerland, Lithuania, Poland, and Slovak Republic. This is another pictures accounting for the annual incidence intervals in Europe, and you can see that this is the area with the highest incidence in Europe. Incidence is a rare disease. Incidence is low, accounting for around 0.063 cases every 100,000 people. So now we know that, according to the other study on cystic echinococcosis, that for every case of alveolar echinococcosis in Europe, we have at least 18 cases of cystic echinococcosis. Highest incidence in Switzerland and Lithuania, and 2 major epicenters with countries around the Alpine area and the southern Baltic area. This is another picture from another study that I'm happy to share with you, and it has been published a couple of years ago and led by my colleague Azzurra Santoro on the genetic diversity of multilocularis in animal and in humans in Europe. And you see, we basically identified 2 major clusters. So I think there could be something in common between these 2 pictures. And also here, I don't have time to clarify the point, but we hypothesized that multilocularis, the genetic diversity of multilocularis in Europe, is a sort of glacial relict, an inheritage from the areas where Arctic foxes were living and were, giving genes of multilocularis to foxes when they recolonized these iced areas. Another picture on incidence in Europe is a snapshot for only accounting only for the last 3 years, and you see that again is a different pictures with incidence in Europe double than before, and the top 3 countries with the highest incidence, again Lithuania, Switzerland, and Austria. And I want also to share another study that we conducted 10 years ago on pooled prevalence of multilocularis in foxes in Europe. And you see that foxes pooled prevalence could be a proxy of human infections in Europe. These are time trends analysis from this study in Europe with observed and predicted number of cases, basically significant increasing trends in all the countries in Europe, okay? With an estimate, according to the model, with around 400 cases in 2024, we predict. And just my last few slides to share with you some news at European level, because as you can see, a new star is born in the field of European public health and parasites, because in 2022, there was a regulation on serious cross-border threats to health in Europe, and that was the consequence of COVID, the pandemic, on surveillance in Europe. And according to Article 15 of the regulation, it was reported the creation of a new European Union reference lab for public health. So there was a specific competitive call in 2024. And it was, according to now the implementing regulation of the Commission, it has been established now a new European Union reference lab for food, water, and vector-borne helminths and protozoa that has been hosted in Rome, in Italy, and it will be operational in 2026. And the focus of the pathogens in the center of this new European lab will be Echinococcus, Trichinella, Toxoplasma, and Plasmodium, sorry. And the mandate for the next 7 years will be to sustain ACDC laboratory disease networks, food and waterborne network, and emerging vector diseases. We will provide the reference material, reference diagnostics, characterization services, and external quality assessment schemes. We support with technical and scientific advice the CDC and the Commission, but also the networks and all the people in Europe needing a support. And we will organize as a mandate the annual meeting and trainings. So next year we will start working on that. And my last slide is that we should also take into account that we are also a European Union reference lab for the parasites in a food safety field, more vet-oriented. So our aim is to establish in the future a new binary star system, keeping together human and animal health in a One Health perspective. And thank you for listening.
Thank you, Adriano. Thank you for this overview and also for this news about this new center. Maybe we can discuss at the end, as Gregory mentioned it. So I will give the microphone to Gerard Yumac. Together with Alessandro Massolo, they will talk about the southward spread of Echinococcus multilocularis.
Yes. I'll just share the file so you will see it.
Yes.
Yes, okay.
Yeah.
Okay, so yes, we have prepared this communication with Alessandro Massolo, a good collaboration, and I will just present you, but we have made this all together for sure. So it will be about the southward spread of Echinococcus metrocularis in Europe. Is it new finding from southern France and Italy's northern Apennines? So I will not give you a lot about the background of Echinococcus metrocularis, especially regarding the disease, as Adriano has give some information already. We think that it's a rare disease, but it's a severe disease. Human is an accidental host, is infected due to the accidental ingestion of parasite eggs. It has a long asymptomatic period of several years, which complicates the identification of the source of contamination due to direct contact with definitive host, but also to others by ingestion of contaminated food, water, also contact with soil, with hand-to-mouth contacts. We have just, by reminding the life cycle, to keep that it is a sylvatic life cycle with the red fox as the main definitive host in Europe, even if other definitive hosts are involved, and the intermediate hosts are the small rodents. So looking at the spatial distribution of the parasite, historically, yes, it was confined to central regions, which we have identified with the slide of Adriano that's around the Alps that we have historical focus. And during the last 30 years, we have identified the parasite in many other areas, many other countries. But how can we really explain this driver of this geographical expansion? One is the fox population dynamics. We have an increase of the red fox population in many European countries. It can be due to the successful rabies vaccination campaigns, a reducing also of the hunting pressure, but also the ecological adaptation of the fox, which has resulted to the colonization of peri-urban and urban areas. So we have this rising fox density, which has, of course, has an impact of increased environmental contamination due to Echinococcus metrocularis eggs. So it can potentially creating new foci of infection when susceptible rodents are present. And of course, more eggs will result to have an increase of the human exposure, and so can result to an increase of the risk of a human infection. The other driver, it's also the translocation and the migration of hosts. We have the movement of infected wildlife, which can, yes, spread the parasite beyond the known endemic regions. Of course, there is no border for the wild animals, and especially the dispersion of young animals at long distances. It is true for foxes, especially as we know that they are more susceptible, these young foxes, to the parasite, are bearing very high worm burden of 10,000 or 100,000 of worms, when the classical burden for adult fox is more than several hundreds. So we have a high part of the contamination, which is borne by small proportion of individuals, which will disperse potentially in other parts and can expand the presence of the parasites. But what about the role of wolves? Their population is also in expansion in both countries, Italy and France. Their diet is not mainly composed of rodents generally, but it can include rodents, maybe more on the young isolated animals. We cannot be included in a pack and be able to hunt some deer or other species. And the main problem is that these young animals infected will disperse at a very, very large distance, especially compared to fox. So it can result to potential leaps of several hundreds of kilometers, and so potentially increase the spread of dispersion of the parasite more than fox can do, which was estimated for fox at a progression of 3 kilometers per year. But of course, with the wolves and the large dispersion of wolves, it can be increased a lot, this speed of dispersion. Another driver of the expansion is also the changing landscape and climate. We have an alteration in the land use, especially due to human practices, regarding agricultural sector, also reforestation and the expansion of green spaces near cities that can, of course, boost the rodent population and thereby supporting EM life cycle when eggs are present and bring by definitive hosts. We have also climate shifts, which may favor not really the survival of the eggs, but the survival and the reproduction of some key intermediate hosts, and so further facilitating the spread of the parasites. But is there really a geographic dispersion or an improved diagnostic surveillance? That is the question. We have a recent improvement in the molecular diagnostic tools, especially with the development of real-time CoPro-PCR. So we have increased the sensitivity of the method. So we have obtained a higher number of detection, and the fact that we can process from feces rather than intestine, it also facilitates the collection of a high number of samples and the possibility to detect low prevalence. We have also intensified the surveillance program in the last decades, and so of course it yields to more detection. So we can reflect both the true expansion and also the better case finding. So it will also helping pinpoint expansion fronts. What is really new about the expansion in Europe in the last decade? Regarding Eastern Europe, we have been informed by detection of new cases in Croatia, Serbia, Bosnia and Herzegovina. But looking more at France and Italy, we have a southern expansion which has been observed in France of the known endemic area with investigation from both rodent and foxes. We were able to detect, you can see this small circle in red here in France, detection in a new department in the Haute-Alpes, which is the southern limit in France. Regarding Italy, we have this single known autochthonous focus in Italy, in the northeast of Italy, in the Trentino Alto Adige region, which was described more than 20 years ago. That was the situation, but recently, if we are looking to France, we have detected an expansion in the southeast of France due to the investigation from wolf feces by CoPro-qPCR. It was thanks to an opportunistic sampling, since there is a monitoring of the wolves population in France, and so we have the feces of wolves which are already available, and we have analyses more than 1,000 which were collected during more than 1 decade, and we were able to have some detection in department as in Haute-Alpes, where we have already identified fox and rodent, as I just told you, but also in 3 other departments where the parasite was not found before and was not even maybe expected, which is department of Drôme, Alpes-Maritimes, and Alpes-de-Haute-Provence, and with quite high proportion regarding the absence of the parasite before. So we are just at the border of the Alps, but looking at the other side of the border, we have the colleagues from Italy which have detected in the imperial regions the presence of the parasite in both wolves and dogs' feces, with another protocol which they have worked monthly in standard pathway and using another protocol of detection based on the detection of eggs by flotation and PCR. And of course, yes, they are detected in both dog and wolves. Just close finally to the case of France, when we are zooming, we can see that we have 5 to 15 kilometers from between the sample in France and Italy, and especially we have some sample which are very at less than 1 kilometer from the border. In Italy, they have recently also detected another place where the parasite is present, which is in the northern Apennine, due to the same protocol, and they have been able to detect in wolves and dogs feces in this 3 and 5%. And so we have this question about this potential eastward expansion of the parasites in Italy from the Alps to the region of the northern Apennines. But what is really the risk to public health? For that, we need to correlate the surveillance finding in wildlife with the observed increase in human cases. When we are looking to the cases previously described in France, in the southeast of France, in the region of PACA, we have only, we can say, 11 cases described since 30 years, according to the National Reference Center for Human Cases. And 5 of these cases, almost half, were in the 2 departments where we have described recently the parasite. We are not able for now to confirm it is really autochthonous or not. We have to investigate a little bit more with the colleagues. And in Italy, for now, there is only one officially detected cases, but it is in Northeast in the previous area identified more than 20 years ago. But it underlines that we have this latent period of several years for development of the disease, so we have to be vigilant to area where the parasite has a low incidence, but also where we have recently detected, or also in area where it is not detected for now, but since maybe we have not investigated for now, that we can have potentially the presence of the parasite in these areas. What are the preventing strategy and the control efforts that we can have? It's really difficult to have some preventive strategies. It's impossible to reduce the movement of the wild carnivores. We can imagine targeting rodents by modeling landscape for human practices, but it seems difficult also. We can hypothesize that we can have some fox deworming strategies, since it is useful to the deworming of fox, but it is only in the short term where we have to maintain the deworming continuously to continue to reduce the prevalence, and it can be done really only locally. So what we have to do is to advocate for the cross-border initiative. So we have a coordinated approach between French and the Italian public health and veterinary authorities. And we need also to have this rapid sharing of the surveillance data from both sides of the border, since it is really crucial, yes, for understanding and trying to contain the parasite expansion. What are the gaps that we have for now? We need to know if it is this presence detected recently, it is local emergence or introduction by migrating hosts. We have an expansion which is suspected to come from the French Alps, going to Imperia and after the Apennines, but for now we have no proof for that. We need to have some genome-based studies, and currently there is the EMSB microsatellite tool that we can use, but it requires a sample of very good quality, which can be difficult to have since we have not adult worms, we have potentially only eggs. So maybe we can imagine to have also new alternative to have some molecular investigation. Another one is to investigate the intermediate host range that we have in this new area. Previously, it was hypothesized that the presence of the parasite in Europe was limited to the presence of Microtus arboris. We can see that probably it is not the case. We can have the presence of parasite in place where this rodent species is not present. So we have to identify which are the species which are locally involved and which is also their susceptibility to the parasites. We can imagine that the presence, the arrival in the next years of the golden jackal may also modify the ecological and epidemiology structure of these areas. So we have to keep that in mind also. And what we have to continue is to try to define the true endemic area of the parasite. So we need to continue this sampling effort, and also, when necessary, to obtain confirmation of the local establishment in fox rodent life cycle, not only the detection in dog or in wolves, especially wolves can have a large dispersion, as I just told you before. So we have to confirm that it is locally present and maintained with the classical life cycle. So what is the home message? Of course, we are talking about that since many years, but this parasite is no longer confined to its traditional hotspot in Europe. We have this range of expansion, which is due to intertwined ecological host population and anthropogenic factors, and international collaboration is paramount. We need to have an harmonized surveillance, consistent communication strategies, and evidence-based control. All of these measures are really key, yes, to mitigating the public health risk and staying ahead of these parasitic threats. The convergence of French and Italian data will also offer powerful insight into how both nations, but of course at a larger scale also in Europe, to better understand and trying to respond to this ongoing geographic expansion of Echinococcus multilocularis. So thank you for your attention. Thank you.
Many thanks, Gerald, for this very interesting presentation. Alessandro, you have something to present too, I think.
Now, we have decided that it will be easier with Alessandro, but is of course available for any questions.
Okay, so how do you want to do? Discussion around Echinococcus now, or?
Yes, I think it's better, Gregory. Yeah.
Okay. Any questions?
I have a question for my friend in Rome, if nobody else has. So in perspective also of these new stars that are born, right? So is there any plan of trying to genotype the parasites that are responsible for human infections? And if so, is this going to be interesting at the European level as it has been, for example, in the case that we studied in North America, where the typing of the parasite allowed us to track down a possible source of infection?
I think that in the future there will be room for such studies within one of these European Union reference lab. The study that I briefly presented on the study of at European level of mitochondrial genetic diversity has been based on both humans and animal hosts for multilocularis. So I mean yeah, the answer is yes. There will be room, and but I don't know at the moment what are the research questions that we should address, because I mean, it's really homogeneous from a genetic point of view, multilocularis, and I mean, I think it would be really interesting whether we can associate diversity with the clinical impact of the disease, if there is any difference in such terms. Otherwise, they will be not really informative.
Yeah, that was where my question was going, whether there is any particular strain within Europe that might be more virulent for people than others, but I'm not sure if that is a working hypothesis that is worth having, because I don't know if there is any previous hypothesis on a differential virulence of different strains within Europe, I mean.
It seems not. Maybe, a study like this will clarify such point, but it's the same for cystic echinococcosis, at least within Echinococcus granulosus sensu stricto. I haven't seen any, correlation between, genetic diversity and virulence and impact in terms of clinical condition in humans.
Thanks.
I have a question for Gerard and Alessandro. I have two questions. First, the weight and the real role of dogs for the spread of Echinococcus multilocularis. And the second is about the convergence of data, French and Italian data. So maybe we can think on something to do together in order to really have a common database regarding these data, but also how to translate the research into practice and policies, in the sense how the flow of communication to the national authorities and also the international one as EFSA or other authorities. So first the doc, then how we can share the data, and the third is how to convince politicians, the Ministry of Health, on the real importance of Echinococcus multilocularius and of Echinococcus granulosus for sure.
Okay,
Because
Gerard.
the official reports are always like 0, 1, or you know better than me.
Gerard, I reply to the dog question. That is the easiest. And then you get the hardest ones, okay? So the dog thing is likely dogs, they are not playing a role in spreading and maintaining the cycle as they do in granulosus, but they may play an important role as a vector to humans. So that could be a risk factor that I would not neglect, but I don't foresee any particular role in spreading, except for some particular situations, intercontinental spread, for example, of strains. But no, I don't see the dogs playing a significant role in the expansion itself. And then I leave the word to Gerard for the other two questions.
Yes, Laura, thank you. I can understand, I can answer to the question also about the sharing data. We are collaborate together, but we have discussed, especially to prepare this presentation, to say we have tried before a little bit, but we have to continue and to have a common project, really to share more data and to make common studies. Since for now, we are sharing our information, but we are doing each other our study in both sides, but it's not exactly the same protocol. So we have to, have both sharing of that, doing the same things, have the same approach, maybe a more global approach on that. And by doing that, we can have hope to be able to have more weight to contact European authorities about this is necessary to do. We can have this nice example of France and Italy, but we can have a lot of example in many other areas in Europe and try to do that. Maybe we can try to do that first in France and between France and Italy, and after use this as an example to try to have a more more bigger project on that and more approach also on that. Just it's not the point about just a project, but just have a more correct approach to be aware of that and to about this expansion.
Yes, I agree with you, and I think that the role, the strongest role is by you and Adriano, because you are in close contact with the Ministry of Health compared to academics and universities. So I really hope that we can do something very, very strong.
After the conversation with Gerard the other day, I immediately called Adriano and I asked,
Adriano is the key.
Adriano, we need you.
Always, we need you. And Gerard as well.
Gerald and Alessandro, I have a very naive question. I don't know about the situation on raccoon dogs in Italy, but in France, it's more and more raccoon dogs. And of course, Echino-multilocularis can be in raccoon dogs. Could it play a role or not on dispersion of the parasite?
To be honest, I wouldn't know. I need to look into it. It depends on densities and dispersal distances, but I'm not that familiar with raccoon dogs situation in Europe, so I can't answer. I don't know if Gerard has more information.
Yeah, with that we know about the population in France, it's still very, very low and very few individuals which are really detected. So for now, it's not really a problem. I can be more prudent about the potential role of a golden jackal. We have few individuals identified, longer probably distance of dispersion, and even the diet is a little different, but I will be more, will not say afraid, but about golden jackal than about the raccoon dogs.
And maybe if we think about hosts, one host that is often under considered, not very much considered, is the muskrat as an intermediate host, because the muskrat apparently is much more resistant than murids so that it usually lives longer with the infection.
Agree.
And it may accumulate the infection and stay in the area much longer. So the muskrat is actually a critical host, very interesting from an eco-epidemiological standpoint, but definitely critical. Yeah, definitely.
May I comment?
Yes.
Of course.
Yes, I mean, I agree because, you know, muskrats are bigger than, you know, the typical, you know, intermediate host. So they can really amplify the biomass of the parasite, and the rodent is spreading in Europe, so it makes all sense. On the other hand, I don't think that raccoon dogs may play a major role. At least during last years, we haven't seen such huge increase in prevalence in raccoon dogs if you compare at country level with foxes. And also we should take into account an ecological, you know, point from raccoon dogs that is somehow not spreading the, you know, the disease, because they use latrines. So they concentrate fecal samples in a single specific point, and this is not favoring the dispersion of the eggs around. And so maybe for that reason, or that reason is maybe contributing to the not high prevalence of multilocularis in raccoon dogs in Europe.
You know, and during the winter, they have hibernation, so they are not very active during all the winter, which is a crucial period also to disperse the parasite. So in Germany, we have data with quite a lot of raccoon dogs, and as Adriano told, the prevalence is lower than in foxes, and they have not observed a really critical important role for raccoon dogs. So yeah, probably not.
And the parasite is from the north, probably. I mean, he really like cold, you know, I mean, maybe it's, you know, it's just an opinion, but one of the reasons why we don't see any huge genetic diversity in multilocularis if you compare with the diversity of granulosus in terms of species that they have within the complex is because we usually see the parasite from an east-west perspective. But this is a parasite from the north. It was probably historically living in Arctic foxes in the north. So that's why if you see from the north of the globe the parasite, you will see not so many genetic difference between North America and Northern Europe, because, you know, the distance are shorter if you see from an Arctic fox point of view the parasite.
Perfect. Any other question? No? Okay, so I think we will go through the program now. Thank you very much again to Gerald and Alessandro for preparing this very interesting presentation, and Adriano too.
I have to say hello to all of you because I have lectures. So thanks a lot for adjusting this presentation at this time.
Thank you, Alessandro.
Thank you,
Thank you very
Alessandro.
much.
Have a
Thank
good
you.
day.
Bye.
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 Bye. Thank you.
So the next presentation will be about the parasite in a fish, and Melanie and Simonetta prepared the presentation, so I don't know who present first.
So
I will share my screen, Simonetta. But go ahead.
pardon, Gregory. I would like just to thank you very much to our society, the Italian Society of Parassitology and the French Society of Parassitology. And it is a very, a really great pleasure to meet you virtually, Gregory, after the IMOP conferences in Poland last August. And I like very much this initiative, even because I believe, I strongly believe that certain parasitological aspects, as those underlined in these talks today, should be should be analyzed and investigated indeed without boundaries. This is the reason why we, Melanie and me, started our collaborative works on one of the most important zoonotic groups of fish parasites, the anisakid nematodes, and most recently even moved to another group of parasites. But I would like just to say that our collaboration started several years ago when Pascal Boireau invited me in Paris at the Hanses to give a lecture on anisakids and anisakid nematodes. And I met Melanie, and she was very young and very enthusiastic to study together these worms, these parasites, and she spent a long time in my lab. So we have the opportunity to work, to start our collaboration, working together. She wanted to analyze analyze and genetically identify some nematodes collected from fish collected from France, of course. But I would like even to say that we have it was just a start, because we have consolidated our collaboration later on in the framework of a European project that we have shared in the years between 2013 2018. Thus it is, and we had the chance, to carry out a large epidemiological work on these nematodes. And even we have the chance to develop new and rapid diagnostic tools. So it is my pleasure to leave the talk to be presented by Melanie, in order that she can present our main results achieved on that, in the framework of that project proposal, European project proposal, but not stopped that, because we are continuously sharing experience, mode of carry out this type of research, and sharing even initiative to submit other European proposals, even recently. So I'm very happy with this collaboration, and I would like to maintain this collaboration with people like Melanie and her team as well. So Melanie, I leave the talk to you. We have prepared the talk together, and Melanie, it's up to you. Thank you. Thank you, Gregory and Laura.
Okay, so as Simonetta, I am very glad, and I thank you to have given us this opportunity today. And as I mentioned, it started quite a while ago, almost 20 years now, and it all started with my stay in Simonetta's lab. And then we had the opportunity to present this preliminary work, in the Viterbo International Symposium on Fish Parasites in 2007. But that was only the start. And before that, I would like to go a little bit into some general data on anisakis and anisakidae. As you may know, anisakids are nematodes, and they have a definitive host in marine mammals. And then the eggs are released in the marine environment, where they hatch to give some larvae. Those larvae may develop into 1 or 2 stages. It's not really clear in the literature. And then at some point, whether the second or the third stage, they are ingested by crustaceans, zooplanktonic crustaceans. And then at that point, there may be several paratenic hosts belonging to fish and cephalopods. And at that stage, either the fish or the cephalopod is eaten by a marine mammal, and then the cycle goes on again, or the fish is eaten by a human, who can be an accidental host. And then some pathology, digestive or allergic, may be developed by the human. So just some example to present to you our common friends. So here are some pictures taken either from the literature or from our own observations. So here on the left, you have, so they are the egg stages, and then the stage that's in the zooplanktonic crustacean, where you can see more or less visible the parasite in it. And then, of course, most of the pictures taken from for anisakis come from fish. And here you have one example of Phocanema in a cod fillet, and one example of anisakis in the liver of a whiting. And the last picture is a stomach from a dolphin taken by Simonetta. If we now take a look at the phylogeny, indeed, all those different genera of Anisakidae are composed of several species. And here is an example of Anisakis phylogeny here, where you can see that so far 10 species have been described. And of course, being, as formerly mentioned, one of the main zoonotic parasite products, at least in Europe, several documents have been produced to assess the risk associated with these parasites, and the most recent ones were produced by the EFSA in 2024, and Simonetta was part of this working group. And so now if we go into some of the results we obtained together, Simonetta's team and my team, the first project which associated both of us was a French project named Fish Parasites, and it was between 2010 and 2014. And the objective was to collect some fish and then analyze them to be able to establish the parasitological descriptors of those fish. And the final aim was to be able to do some risk assessment. For this first project, we analyzed, so not only us, but there were several partners on this project, but we analyzed 1,700 fish belonging to 18 species. And one of the, so as you may see on the map, we had samples coming from either scientific campaigns that are labeled as small fish, or from professional fishermen that are labeled as anchors. As you may see, we have samples mainly close to the French fishing areas, but also in some larger parts, but from French fishermen. One of the main results we obtained from this project was the differences obtained in prevalences between those fish species. As you may see on this graph, we had prevalences going from 100 to 0, depending on the fish species. And also we obtained very different prevalences between the whole fish and the fillet prevalence. And this is one of the results that's very interesting, since it's very different to have parasites within the viscera or within the fillet, considering the food safety aspects. So, and then, as Simonetta mentioned, she very kindly invited me to participate to the parasite project that was carried out in the mid-2010s, and it was coordinated by the CSIC, and many partners were involved. And some of the aims of this project was to do a major epidemiological study, and also to be able to identify the hazard and to give, again, some data on the prevalences and the identification. So with Simonetta, we mainly worked on those two work packages, work package 2 and work package 4. And so once again, it was not only the two of us, but during this project, more than 17,000 fish were sampled during 2 years belonging to 16 species. As you may see here, the different numbers. And the fish were sampled in different fishing areas, all from different stocks. And it was the biggest ever epidemiological study on fish parasites by the number of samples and the geographical areas in Europe. So now I will present to you some of the results we obtained during this parasite project, and also related it to the publications where we were both associated. So the first publication was just a global view of the survey, of the epidemiological survey. And also one of the main points of this survey was that we were all using the same method to detect the parasites, and it's the UV compression method, as you may see an example on this slide. And the advantage of this method is it's exhaustive, so you will see any one parasite present in the organ, but you may also be able to describe precisely where the parasites were. As you see in this picture, you may see that the parasites were not regularly distributed between the different parts of the fillet, with the majority of the parasites present in the belly flap, so the ventral anterior part of the fillet. But I will come back to that. So some of the work we carried on was made on cod, and here you have some of the results we obtained in this paper, and we described the presence of several Anisakidae and Rhaphidaskaridae parasites in cod, and we also observed very strong differences in prevalences between the fishing area. I'm sorry, I missed some of the areas, but those were the prevalences for anisakis in the Barents Sea, in the Baltic Sea, in the northern North Sea, and in the central North Sea. And as you see, we have two different patterns with very high values in the northern North Sea and the Barents Sea, and very low values in the Baltic Sea and the central North Sea. Still on the cod sampling, we also analyzed several factors. So as I mentioned earlier, the fishing area was one of the factors, and the other factor we analyzed as being significantly influencing the prevalences and intensity was the length and the weight of the fish. As you may see on this graph, here is the total length of the fish and the total weight of the fish, and here the intensity of intensity of parasites in the fillets. And you may see that we do have a strong correlation between either the length or the weight and the number of parasites. Since on the cod, we analyzed also the distribution of the parasites within the fillet, and to establish whether there were differences between the left and the right fillet. And as you may see on this graph, there was no significant differences between the left and the right fillet, whereas we observed strong differences between the anterior part of the fillet. So if you see on this picture of a fish, and that's not a cod, sorry, it's a mackerel, but this is the anterior part and this is the posterior part. And we did find the vast majority of the parasites being present in the anterior part or present in the ventral part of the fillet. And this is quite relevant with former data and also with the picture of the UV press method I mentioned earlier. We also did some work on the mackerel, and this is one of the data we obtained about the overall abundances of Anisakidae Anisakis parasites, in the different sampling areas. So the North Sea, the Norwegian North Sea, the Faroe waters, the English Channel, the area of Spain and Portugal, and the Mediterranean Sea. And as you may see again, like for the cod, we had very strong differences between the areas. And also during this project, we also worked on several other species like the herring and some flatfish and some others. But I would like also to mention to you one of the important work that was carried out during this project, and that was the development of a multiplex qPCR species-specific technique that allowed to identify fish parasites of the different genera, either Anisakis, Pseudoterra nova, and also Hysterotilacium. And as you may see here, that in one multiplex qPCR reactions, we were able to identify all these different species with different probes and primers systems. So that's something that really allowed to identify a lot of individuals, because before that it was needed to go through the Sanger sequencing step, and that takes a lot of time and may be quite expensive. Whereas here we managed to develop a rapid and low-cost technique with a high sensitivity, a high specificity, and also reproducibility. And this is one of the tools that we are still using together today. And so, of course, for a long time, we had a long time and fruitful collaboration on anisakis, and this is still going on today, as Simonetta mentioned earlier. But also most recently, we started to work on other parasites, such as Cryptococci lingua. So Cryptococci lingua is a trematode that may induce black spots on fish, as you may see on this picture here. And if we look closely at those black spots, it's a metacercaria of a trematode. And the cycle of this trematode goes through epicerose birds that will release eggs in the bird feces. And those eggs will go inside some snails, where they will evolve within 4 successive larval stages, until a free cercaria is emitted in the water. And this free cercaria will go and actively infest some fish, where the larvae will, where the cercaria will transform into a metacercaria. And I'm sorry, I'm missing a arrow here. And then once the fish is eaten by the bird, then the cycle goes on again. This parasite has very few data available, and if we look at the literature, it is considered as zoonotic. However, there are very few data on this, and we started to work on this parasite some 6 or 7 years ago in the lab to try to elucidate some elements. Among the elements we tried to work on was the population genetic relationship between those different parasites. And this led us to publish this common work with Simonetta's team. And among the work present in this publication was the haplotype network results, either depending on the area where the fish were sampled. So here on the left side of the haplotype network on the left side, you have the sampling coming from the English Channel or the 1000 bight of the North Sea, the East North Sea, the Central North Sea, and the West North Sea. And as you may see on this result, we did not manage to identify any haplotype related to the geographical areas. And we had the same result with when we looked at those parasites sampled in different fish, because the fish that were sampled were whiting, put, dab, sprat, herring, plaice, and flounder. And again, we didn't find any significant differences in the haplotype networks between the fish species. So this is it. This is what we've been working on briefly over the years. And I would like to thank you for your attention. And of course, I would like to thank all the people who worked on those different projects, whether they were in Simonetta's team or in my team. Thanks to you.
Oh, thank you very much. And so we can start our discussion. Very interesting, your talk, as usual. Is there any question? Maybe Gregory?
Yes, I have
Yes.
a question. Melanie and Simonetta, you showed different data on the contaminations of the fish between Mediterranean Sea and Atlantic Ocean. Do you have any reason why?
I guess for the Mediterranean, it would be better if
I go.
Yeah.
I go first. Well, one, I mean, the fishing grounds have been identified, I mean, just as one of the main results of this project. as one of the most important driver in the epidemiology and of these parasites. I mean, the infection rates in fish, and generally of the density of the parasites in different fishing grounds. This reason is mostly related to the population demography of their definitive host, and especially definitive hosts, which are those which are responsible for maintaining the high population demography of the parasites in a certain geographic areas. I mean, different aspects related indeed to the definitive host, and even population, different stocks of fish from Mediterranean area, which is different from the Atlantic areas, would be responsible for this level of a different level of infections. I mean, generally of the demography of the population sites of the parasites in different fishing grounds. We are also in this last period, we are also correlating the population sites of these parasites to their genetic variability. I mean, values. I mean, their genetic polymorphisms. Indeed, we have seen, for instance, that there is a positive correlation between the density of the density values of the parasites and the genetic polymorphism of these parasites. Thus, for instance, the population of anisakis, for instance, from the Mediterranean area is at a lower level of demography, but also at a lower level of genetic variability with respect to the populations occurring in northeast Atlantic areas. So these aspects, in our opinion, I mean, me and Melanie, indeed, we are working also on these aspects. We are complying, indeed, samples, material, because this type of analysis requires a lot of statistical elaboration. And but one of the drivers is just that, it's linked to the demography of the parasites, linked to the demography of the host involved in the life cycle of the parasite. Maybe Melanie would like to add more.
It's just something that I haven't presented today since it's more recent work, but also we are now working on the feeding habits of the different fish, depending on the different geographical areas. And we do find some differences. It's at more local level, so it will be between the English Channel and the southern North Sea. But we do have strong differences, and we managed to relate them to what the fish eats. And the fish also has a different, of course, feeding habits depending on what it can find in the environment around it. So indeed, it's, as Simonetta said, it depends on the host population distribution.
Okay, thank you.
Thank you, Gregory, for the question.
Any other question?
Maybe we can thank Melanie and Simonetta.
Oh, thank you
It was very, very inspiring. And you know that in Italy there are a few groups working on fish parasites. and Simonetta with her group is one of the most important group, but also together with the group in Padova and the group in Bologna.
Yeah.
I say here,
And
yes, Monica Caffara.
Yes.
And Monica, yes, Monica is one of the organizers of the meeting in Bologna. So together with Andrea Agustinelli, Maria Letizia Fioravanti, Roberta Galuppi. And so I think there will be a big space for fish parasitology during the meeting in Bologna. And thank
Of
you
course.
for,
Yeah.
we
for
We
do.
will do
organizing.
it. Absolutely.
Yes.
But I see also Gabriella Gaglio
Gabriella
from
Gaglio,
Sicily.
I didn't see her.
Yeah.
Okay.
She's also working from Sicily, working in the
Yes.
fish parasite.
Yes, Gabriella, ciao. Sorry.
Ciao.
I have two
Ciao,
pages on
Gabri.
my Zoom, so I didn't see.
See, indeed.
Yeah.
And Federica also is online.
Yes, Federica from
Marcel.
Padova.
Fede.
Yeah.
Indeed.
So we will do our best to give a huge space for fish parasites.
Yeah.
Yeah. Also for the other one, but fish
Yeah,
parasites.
but fish parasite deserves because it's a bit a niche. It's very important, but not lots groups work, at least in Italy. So you are really the reference labs.
Okay.
So, and I take the opportunity also to say that the Parasitology Research Journal, as in which for which Federica, Marcel, and me are section editors responsible for the fish parasite section. I has agreed to give us the opportunity for a special issue devoted to fish parasites in the Anthropocene. The idea was started during the last conference, international conference on fish parasitologists in Merida, where also attended by Monica, by Melanie, by me and our teams. So they accepted the idea. So I invite you all to contribute to the special issue, and we are all together. It is a good opportunity to produce something together, some of our shared interest. So I would like to thank you to Laura and Gregory for this. All the board of our society, without boundaries.
And Gregory, I come back to ICOPA in Canada because I forgot mentioning that Simonetta and Marco Lalle are in the board of the World Federation. Yes.
Yeah.
And so I think this could be really an occasion to propose a joint session. I think this will be great.
Yeah. I think that we should start from here. From the strategy to be without boundaries in Europe, to propose something at the world level. And if we share interest, it is the moment to do that. And to be, they accept in the last meeting that we had last week. They are opened to symposia, symposia proposal. So I think that a good example should be to be like a unique society from Europe, and to try to do something together in this sense.
And the call will
without
be
no competition, so without no strategy, just to propose something. And another things that they would like to stress should be to give effort for the young participation to the ICOPA. and the society should make some support in doing the yes, they would like to receive some support, from the single society for this support for young people attendance ICOPA. I think that it will be expensive in any case.
Monica, you want to say something?
just a few words, because back from Fish Parasite, I want to tell to all the SoIPa friends that in 2029 we will have the International Symposium on Fish Parasite in Bologna. We win again.
Great.
So
Exactly.
please help us also in organize this meeting. Me and Melanie are part of the international committee. Simonetta was part of the international committee for a long time. And so, yeah, all of you are invited. But I will remind you, don't worry.
Yes, and we will have the assembly in June, so we can remind everybody, all the members.
Absolutely. Okay. Thank you.
You're welcome.
So I think that also they did a good initiative. I will send you Laura and Gregory or Melanie as well. Have you seen the trends in parasitology notice related to the last symposium in Mexico?
No.
They have produced a summary of all the contributions, including yours, Melanie, Monica, and all of us as attendant people. So it was a nice initiative, and it is nice. I will share with you, Laura, and with you, Melanie, so you can pass to Gregory or Supputo in our websites. So thank you.
No, thank you. And Gregory, I think we should work together not only on research, but also on teaching. And Simonetta is here, she coordinates the teaching committee, because we have really to push all the faculties, the veterinary faculty, the medical faculty. Yeah, I don't know, maybe in another roundtable we can discuss about the situation of teaching parasitology in France and in Italy. I think this would be great.
When I introduced the French Society activities, I could not speak about everything.
Yes, of course.
Of course.
For example, you mentioned the summer school you propose on entomology, for example, and Émilie Boussira does the same in France. So maybe it's a way to collaborate. I don't know. Probably the people know each other already, but maybe not. We don't know. Maybe we can.
Yes, I think that the best could be that especially our young researchers collaborate, and I'm sure they already collaborate. I know that Lavinia, for example, my researcher, collaborates with Emily, and so I really invite all the members to do what Simonetta, Melanie, and Gerard, Adriano, and Alessandro did. So we can really plan a regular, for example, every 6 months webinar in order to discuss and in order to share information about also policies.
Yes, sure.
Yeah. So Adriano,
good.
You turn on your camera, so you will be one of the main promoter of this collaboration.
Good.
You're on mute.
We cannot be on mute, Adriano. Yeah.
Okay, thank you. I will be happy to do that.
Yeah. And I will meet Adriano and Gerard very soon here in Napoli for a meeting on Echinococcus granulosus, so we can share again some information and some plan to go ahead. And I say hello also to Frank. Where is Frank? It's a pleasure.
Thank you, Roa.
Ciao,
Thank you.
Frank. Ciao, we miss you. And Gregory, I think it was a success because we had the 51 people, and we know that now, this is the teaching semester starting. Everybody is really involved in so many meetings. So I think it was really a success.
Yeah, I'm very happy with the participation of both members. So yeah, it's successful. Yes.
We have to continue. We should continue.
Well, we promised to announce it a bit before because
it was just a last minute.
Very late this time, but it was the first one, so we are very happy to have pushed to have it. So next time, of course, we will announce ready earlier.
Okay. So thank you very much to Simonetta, Melanie, Adriano, Gerardo, Alessandro, and for all participants. And we keep in touch with the joint collaboration.
Keep in touch.
Yes.
For sure.
Yeah.
Thank you.
Ciao.
Thank you. Ciao.